Hey guys, welcome to Biomix Lab. In this video, we are going to discuss about the regulation of the cell cycle. So what happens when the cell is present? It needs to divide itself into two daughter cells. Whenever it is dividing, whatever is present in the parent cell, it needs to transfer into the daughter cell. Like in the cell, what is present? There are different type of cell organelles as well as the genetic material is in the form of a DNA. So this thing needs to duplicate itself before it is dividing into two daughter cell. Then only it can be transferred into the daughter cell. So for that, the first thing which a cell needs to do is its growth. The cell has to increase its size with the growth. The second thing is the chromosome replication because the genetic material which is present, it is in the chromosome. So it needs to make a copy of itself. So now two DNAs are present by the help of replication. Now they needs to segregate. That means they needs to be present in two different parts. Like here we can see that now this is a cytokinesis phase. The chromosome is segregated and this is a cytokinesis where the division has not taken place. Only the cytoplasm is dividing here. After it has segregated, the cell will now divide into two daughter cells. So when a cell is dividing, it is entering into different phases of cell cycle. The first one we can tell as interphase. This interphase is again divided into three parts like G1 phase, S phase and G2 phase. G stands for growth. We can also tell it as a growth 1 phase. S is the synthetic phase or synthesis phase. G2 is the growth 2 phase. Next comes the mitosis. Mitosis is also known as M phase. So what happens in the G1 phase? In the G1 phase, the cell is ready for its growth. That means growth is taking place here. That means the cell will enlarge itself. As well as whatever is required in the S phase, it is synthesized. Whatever proteins are required in the S phase, it is synthesized or it is produced in the G1 phase. But the proteins are in the inactive state. When the cell enters into the S phase, then only the proteins will be activated. Now, when the cell is entering from the G1 to S phase, this phase is known as, this part is known as the transition phase. This is known as transition phase. So the cell is having different transition phase, like when it is entering from G2 to M, it is known as G2 to M transition. When it is entering from metaphase to anaphase, it is known as metaphase to anaphase transition. So now when the cell has replicated its DNA in the S phase, the replication has taken place. Then the cell enters into the G2 phase, that is the growth 2 phase. So what happens in the growth 2 phase? Whatever things are required in the metaphase or whatever proteins are required in the metaphase, it is synthesized in the G2 phase. As well as if the DNA is synthesized and it has entered into G2 phase, the G2 phase will check whether the DNA has properly replicated or not. Then only if it has replicated, it will transfer it into the M phase. Okay, so when it is entering, when the cell is entering into different phases, it is having different type of checkpoints. So what is the work of this checkpoints? The checkpoint decides whether everything is proper or not. Like in the G1 phase, what was taking place? In the G1 phase, the cell was increasing its growth. So this checkpoint checks that whether the growth has taken properly or not. What was the other thing? The other thing was that production of the proteins or the enzymes which are required in the S phase. The G1 checkpoint checks that whether all the proteins which are required in the S phase, they are synthesized or not. This is the work of the G1 checkpoint. Next comes the G2 checkpoint. In the S phase, the DNA is replicating, then it is entering into the G2 phase. This G2 checkpoint, what it does, it checks that whether the DNA has properly replicated or not, whether the tubulin has been synthesized or not, whether there is proper ATP or not. It will check because this tubulin is required in the metaphase. Why this proper DNA replication is required? Suppose a DNA is present and it is having some mutation. If with the G2 phase, this is not checked that whether the DNA is properly present or not, whether there is any mutation or not, then this DNA will be transferred into the daughter cell, which will create problem or it will lead to some of the diseases or some of the abnormalities. That is the reason these checkpoints are present, which will help in the which will help in checking whether everything is correct or not. If there is any problem, then the repair mechanism of the cell will be activated and the DNA will be repaired. Then the cell cycle will enter into the next phase. Next comes the M checkpoint. What happens in the M phase? In the M phase, the chromosomes which are present, they are placed in the middle. Okay, they are in the middle. Why they are present in the middle? Suppose this is a cell. In the metaphase, these chromosomes are present in the middle because now in the anaphase, the segregation will take place like this. Like one part of the chromosome will be transferred into the one pole, the other will be transferred into the other pole. Then the cell will divide and it will have equal amount of chromosomes or the DNA. So this will be checked by the M checkpoint, that whether the spindle fibers are present or not, if it is present, if they are attached to the kinetochore or not. So when this cell is going into the cell cycle, there are some proteins which are regulating them. What are the proteins? The proteins which are regulating the cell cycle are the cyclin and the CDK. So this is a complex and this complex is known as MPF, that means maturation promoting factor. So there are two things, that is the cyclins and the CDK. The cyclin which is present, it is a regulatory component, and the CDK which is present, it is a catalytic component. So what is this CDK? It is cyclin dependent kinase. That means it is a kinase, that means it is phosphorylating something and it depends upon the cyclins. There are different type of cyclins as well as CDKs in different phases of the cell cycle. Like there was G1 phase, G2 phase, S phase, M phase. So in each phase there are different type of cyclins as well as CDKs. So there is destruction of the cyclin in every phases of the cell cycle. Like in G1 phase, if some cyclin is present, it is working in G1 phase, but it will not work in the S phase. So during that time, it is destructed and another cyclin is synthesized. But the CDK is constant. It is not destructed, or it is regulated by the phosphorylation and the dephosphorylation. So let us see how this CDK is activated. Initially, it is in the inactive form. How, if it is activated, how it is activated, let us see. So this is an inactive CDK. The CDK is having a T loop with it, and the T loop is arranged in such a way that it is not showing its activating site. But when the cyclin is entering, or when it is combined with the cyclin, it is showing some of the conformational changes, and the active site is visible now, or the active site is exposed now. By the help of another kinase, that is a CDK activating kinase, this T loop is phosphorylated. The activating site of the T loop is phosphorylated because as we know that the kinase is helping in the phosphorylation, it is being activated, and now the CDK which was in the inactive form, now it is in the active form. Okay, so the cyclin and the CDK, when it is present, they are regulated by two things. What are they? One is the activating kinase, one is the inhibiting kinase. The CAC which was present, this CAC is the activating kinase. When it phosphorylates the CDK, now it is active. The other one is CKI. The CKI means CDK inhibitor kinase. The cyclins, as we have discussed, that cyclin is destructed, but the CDK is not destructed. How this, if the destruction of the cyclin is taking place, how the cyclin is being destructed? It is destructed by two components like the APC and the SCF. So the APC which is present, it is activating by one of the activating subunit that is known as CDC20. And this APC which is present, it is degrading the cyclins of the M phase. Like we can see that now this was an inactive APC. When activating subunit that is CDC20 is combining with it, it is activating the APC. complex. Now this is in the active form. Here what we are having a CDK as well as M cycline complex, which is known as maturation promoting factor. Now the work is that we have to disrupt the cycline. This APC, it is a ubiquitination protein. So what it does? It add the polyubiquitination. That means multiple ubiquitin proteins are being added. So now the M cycline which was present, it will be disrupted and it will not be functional. The next is the SCF. How this SCF is regulating? So when we are having a CDK when we are having a cycline CDK complex, now it is in the active form. Now it is in the active form. This there is a kinase, that is the CKI. CKI is present. This is a CDK inhibitor protein. What the CDK inhibitor protein does do is it will not allow the cycline and CDK to work. So when a cycline needs to work, what it will do? It has to remove the CKI or the inhibitor protein. So this SCF which was present, it is also a ubiquitin ligase. So the work of the SCF is it will add polyubiquitin into this protein and it will help in the destruction. Now the cycline and the CDK will be activated. We can see that here the CKI is present, which is phosphorylating itself and it is activating. So this CKI needs to be deactivated or it needs to be degraded. It is degraded by the SCF complex by using the polyubiquitination chain. Okay, so the APC which was present, it is activated by two factors, like one is the CDC20 and the second one is the CDH1. When it is activated by CDC20, it works on securin and it allows the cell to go from metaphase to anaphase. Okay. Now, when it is activated by CDH1, it works on a mitotic cycling. That means when the anaphase is present and it has to convert into G1 phase, this APC is activated by CDH1. Okay. So when the securin is present, it will not allow the chromosomes to separate itself. Okay, so when it is restricted, it will allow the metaphase to, in the metaphase, it will allow the chromosomes to separate. This was the work of this APC. Now comes the cycline and the CDK. As I have already told you that there are different type of cyclines as well as CDKs in different phases of the cell cycle. So the cell cycle is divided into 4 phases. That is the early G1 phase. And in the early G1 phase, the cycline which is involved is cycline D, CDK4 and CDK6. The CDKs are CDK4 and CDK6. The cycline is cycline D. In the late G1 or early S phase, the cycline which is involved is cycline E and CDK2. In the S phase, the cycline is cycline A and the CDK is CDK2 and CDK1. In the M phase or G2 to M phase, the cycline is cycline B and CDK1. So let us again see. For G1 phase, it is cycline D, CDK4, 6. In the G1 to S phase, it is cycline E, CDK2. In the S phase, it is cycline A, CDK2, CDK1. In the M phase or G2 to M phase, it is cycline B and CDK1. Okay, D, E, A, B, 46, 22, 11. Okay. In this diagram, we will see that there are various types of peaks of cyclines as well as the CDKs. Here in the first picture, we have peaks of cyclines. In the second picture, we have the peaks of CDKs. How these are regulated? So what is a G1S cycline? The G1S cycline is a cycline E. So where it is activated? It is activated in the G1 phase, and it is in the peak in the G1 phase. And until S phase, until the starting of the N phase, it has been synthesized. In the S phase, it is disrupted. Now comes the S cycline. The S cycline means cycline A. Okay, it is cycline A. The cycline A is synthesized in the late G1 phase. In the late G1 phase, it is synthesized and it is constant throughout the S phase as well as the G2 phase. In the mid of the G2 phase, it is destroying itself. And when the cell enters into the M phase, there is no cyclin A present, or the S cyclin is not present. In the similar way, now about the M cyclin. The M cyclin is cyclin B. The cyclin B is activated, or it is starting its peak in the G2 phase, in the starting of the G2 phase, and it is in the peak in the M phase, late G2 or M phase, and it lasts till, and it is constant in the M phase till the M phase until the cell is ready for the anaphase. Okay, this was something about the cyclins which are involved. So there are also CDKs which are involved. Now we can see that the G1S CDK which is present, it is activated in the G1 phase. The S CDK which is present, it is activated in the starting of the S phase. The M CDK which is present, it is activated in the ending of G2 phase or G2 to M phase. Now this is a graph of the CDKs. See, here this was the M phase CDK which is present in the G1 phase, and the CDK which is present for G1 to S phase, it is in the peak in the G1 phase. In the G1 phase, we can see here. The S phase CDK which is present, it is synthesized in the G1 phase, and it is stable in the S phase until the mitotic phase. Until the mitotic phase, it is stable and it is degraded in the mitotic phase. Now comes the mitotic CDK. This mitotic CDK which is present, it is activated in the G2 phase, and it is still the G1 phase. Here we can see that here you were having the G1 phase, mitotic CDK was present, so this was the elongation of this peak. When everything is ready, the APC, that is anaphase promoting complex, is also activated in the mitosis phase. This was a graph of the cyclin as well as the CDKs which are activated. So the CDKs and the cyclins which are present, they are different in case of a vertebrate as well as in the yeast. In case of vertebrate, what we have discussed that there are some phases like G1 phase, G1 to S phase and M phase. The cyclin which was involved are, in the G1 phase it was cyclin D, G1 to S phase the cyclin was E, in S phase A cyclin, in M phase B cyclin. And the CDK which was involved was 46 for G1, G1 to S2, S phase 2 and 1 CDK, in M phase it was CDK 1. This was for the vertebrate. But in case of budding yeast, the CDKs which are involved is CDK1. Only one CDK is regulating in all the phases of the cell cycle, but the cyclins are different. Like for G1, the cyclin is CLN3. For G1 S phase, the cyclin are CLN1 and CLN2. For S phase, the cyclins are CLB5 and 6. For M phase, the cyclin are CLB1, 2, 3, 4. So the questions which are asked from this part are like, what is, if we are having the invertebrate, the cyclins are 4, 6. What it is for yeast? The answer will be CLN3. In the same way, if they are asking for S phase, what is the cyclins involved in the budding is for S phase? The answer will be CLB5 and 6. Okay. So this was the difference between the cyclins and the CDKs which are present in case of vertebrate as well as in case of a yeast. So these are the checkpoints. Here we are having the checkpoints which are involved during the all phases of the cell cycle. So how this checkpoint is regulating? If there is no favorable environment, if the cell cannot grow itself, the G1 checkpoint is regulating it. Now the cell is indicating that the DNA is damaged. So it will inhibit the G1S CDK as well as the S CDK. If there is no replication of the DNA, if anything is damaged, then it can also inhibit the M CDK. If the chromosome, if until M phase everything is ready, like there is proper DNA replication, everything is ready, but in the M phase the spindle fibers which were present, it cannot bind to the kinetochore. During that phase, the anaphase promoting complex is being inhibited. So about the basics of the cell cycle regulation. In the next video we will discuss how the cell cycle it is Regulating the G1S transition. What are the factors required? How it is regulating?